Welcome everyone. This is Research Hangout. I think there are some new faces around and some people I can recognize from other editions. So for the new ones, this is a event we run every 2 weeks, and it's basically an opportunity to connect with some researchers from Cohere and Cohere Labs. We discuss different papers, different topics. So if you are not part of the Cohere Labs Discord, feel free to apply to that. I think you can Google Cohere Labs Discord, and there's the invite somewhere there. But I can try to share the link later on the chat. And yeah, today we're going to be discussing bioreason, this paper from Arib. I was taking a look this morning, and I think I understood some parts of it. Some others I didn't. So expect many questions from me because I have a lot of questions. And yeah, just so people know, feel free to raise your hand or post on the chat at any point, and I will keep track of that. And yeah, I think Adi will be happy to answer everyone's questions when you have them. And with that, all yours, Adi.
Yeah, thank you so much for having me. Today we'll be talking about bioreason, incentivizing multimodal biological reasoning within a DNA LLM model. I think it's been a great work of me, Andrew, Purav, Rex, Jack, Arnav, Han, and Omar, Hani, Chris, and Bo across different institutions from Toronto to San Francisco within Cohere and Middle. And I think the whole goal of this work is to say, can a language model learn to reason in biology, looking at a DNA sequence and trying to predict whether that mutation is going to be leading to a disease, and if so, how it leads to. And when you're training these models, some unexpected things might happen, right? So you might pass in like a genetic pathway and say, okay, there's a mutation here. Can you tell me what's going to be the effect of this mutation? And sometimes we'll just spit out Guyanese to you, or it might say, I'm sorry, LLM fine-tuning could be sometimes a bit unexpected. You could interesting engineering that goes into making this thing work across biology and the everything we know in NLP. But ultimately, the architecture looks something of a combination of a large language model, the DNA foundation models, and reasoning coming together. I'm going to break down what each of these parts are doing, and then we can see how all of them work in the final space. So I guess we're all familiar with LLMs. They're essentially big autocompletes. They can take in a sequence of tokens and give you the next token after. But fundamentally, what an LLM is doing is it's learning embeddings from a sequence of tokens and contextualizing those embeddings such that it makes it easier to look at the final words embedding and predict the next word. So these are more like giant embedding machines, and we're just using them to predict the next word, but we can do many things with them. And embeddings are cool because they'll allow you to now go from just a generic set of embeddings of some tokens to now more contextualized, more nuanced understanding of what that sequence means. On the other hand, we've applied the same logic to DNA. DNA is also a sequence of characters like ATCG, and we could do the same auto-regressive modeling that we do with language on DNA, where we create DNA foundation models like EVO2. Here, the model will just learn to predict the next nucleotide, whether it's A or T or C or G, given the previous sequence of all these characters. But the benefit that this approach has is that you're going to be training this model on hundreds of billions of different DNA nucleotides. And as a result of that, the embeddings that you get from DNA is much higher quality. From a language model, you can also kind of pass in ATCGs and get some embeddings out of it, but those embeddings are not nice. The language model has probably not seen much DNA sequences for training data, so it doesn't really have an idea what a mutation could look like, what different parts of the DNA sequence mount to, and that's one of the reasons biologists train foundation models for DNA or protein or RNA, because they want to capture the full breadth of all the information that modality could have within these models. And ultimately, we have reasoning, where you're going to be generating answers to a question, but each time your generation is slightly different, which leads to different answers. And by seeing which answers are more correct than others, you might get a sense of how to think better in order to solve more of these questions right in the future. And the good thing about reasoning models is that with a small amount of data, they can essentially augment their training data to look for the patterns and look for what actually makes a successful answer by just trial and erroring the different types of generation possible. And this will help you augment your capabilities when the data is limited. So putting all these things kind of together will look like something like this, which is like a very big architecture, but we're going to start breaking it down. We'll start from here, where we have a bunch of DNA sequences as inputs. Essentially, you can think of these DNA sequences as images, similar to how a vision language model will take in images and embed them and then put those embedding in its context to be able to process them. Here, the DNA sequences will be passed to our Evo2 DNA foundation model. And this model is frozen, but it will give you very high quality embeddings of that DNA sequences. And then those will be passed on to the context of a language model. Along with the DNA sequence, there's some question, like here saying, oh, there's a genetic pathway, these are all the genes involved, and we know that a specific gene has some mutation, what's the effect, how does it lead to a disease? And this question will also be tokenized and embedded, and we put all these question embeddings in the context. Now, the DNA sequence has the mutation. You can see like from C to G, there has been a change. And we hope that the DNA foundation model has seen so many different DNA sequences that when you pass it these two different DNA sequences, its embeddings will capture the effect of what that mutation is, how it might affect the sequence, how it might affect even the protein that comes from that gene. All those information is kind of locked in within those embeddings. And the question will carry out information about what's the genetic pathway. What do we know about this gene? What do we know the disease that this pathway might be involved in? All those high-level biological knowledge. And so when you bring both of these together, you're kind of uniting the low-level biology of the mutation with the high-level biology of understanding the disease and the genetic pathways. And you'll be passing all these embeddings to Qwen3, which here is a reasoning model. And we'll think step by step of how that mutation will cause a change in the protein, causing the change in the pathway, all the way to what we know about the biology that will lead us to a final disease outcome here in Parkinson's. And you can do a bit of SFT to kind of get this thinking process started, and then you can do GRPO on top of it to allow the model to explore different ways to think biologically and see which ones are more plausible and lead to better diagnosis of the disease. But overall, the key perspective here is you could have done all of these things with just Qwen. There was no need for DNA foundation model. You could just pass everything, all the DNA, all the questions into Qwen. But you won't get the best result because Qwen has never seen DNA sequences. And so being able to leverage a DNA foundation model to give you higher quality embeddings and then having Qwen actually learn those and be able to leverage them for downstream tasks of disease prediction is ultimately what makes BioReason work much better than all the baselines out there. We can also put into a bit more math-heavy way of looking at it, where you have a DNA foundation model, fDNA, we will just take in a bunch of input genomic sequences, and it will just map those to a bunch of embedding vectors. And on the other hand, you have also your language model, which will just take a bunch of tokens, which we call static query of it, and we'll just try to output some text that will ultimately be used for just comparing to ground truth and changing its parameters. And in order to integrate these two, we kind of need a projector layer that will map the embedding space of the DNA model, that whatever that embedding dimension is, to the embedding dimension of the Qwen model, so that you could actually put those into context. And this could be just a simple linear layer, it could be a two-layer MLP, it doesn't really matter. It will just be a projector layer trained with the train, and then we also do a bit of LoRa on the LLM. So that's how your parameters come in, but the DNA model is frozen. So we just look at whatever the DNA model already has learned, and we try to just get the most out of it using a large language model. All these embeddings will be put together into the same context, passed on to the language model, and the language model's ultimate objective is just autoregressive on the data we prepare. So putting this architecture aside, then the question becomes, well, how do you also do GRPO on it? And to be honest, we just look at the most simplest way to do it, which is essentially just take what works in R1 and just do it here. Here, the rewards are just a correctness reward of whether you get the final answer right, and a format reward, just use the right format. And use everything else the same as what the R1 paper did. Looking at the data, I think, is the most important part of this project, because ultimately you need to teach the model how to think biologically. And in order to do that, you should go back to see how biologists actually think about this in the first place. So there's a database called the KEGG database. And in KEGG, essentially, we have the same type of data we've seen before. There's a bunch of genetic pathways with information about those genes and a disease that we know a mutation in some of them will cause, and we know the exact mutation. And some biologists has kind of sit down and written like a paragraph of ground truth of how that mutation is leading to the disease based on understanding of biology. This is your verified, explanation of the biology, and so we can trust that this makes sense. We'll just pass this to Claude and just ask it to put it into question answer format, kind of breaking down this paragraph of ground truth into more like step-by-step reasoning. So ultimately, your data will be looking like this, where you have question with like the mutation, the actual reference, and the variant sequence. The chromosome number, the pathway, the information about those genes, and then asking a question of what's the disease that will lead to. And the answer will look like a step-by-step thought process of going from just how the substitution changes the protein all the way to the ultimate effect in the biology, and then kind of giving us a summary and a final answer prediction here in Parkinson's. At the same time, you can also look at the diversity of cake. Like it's about 1,500 question answer pairs from 173 networks and 37 diseases. So it's not that big of a data set, but it gives you enough data points to at least train that projector layer and ultimately do a bit of GRPO. Now, the results here are quite surprising. Like Bioreason boosts performance by quite a much, and it essentially solves this benchmark. And it's quite nice to look at the different base models we've trained and also the actual Bioreason model and kind of compare them to see how different gains are acquired. So one way to do this task is to just use a DNA model. Like you can just pass in the DNA sequences we saw to EVO2 or nucleotide transformer, both state-of-the-art DNA models, and just take their embeddings and maybe do a classification on top of it on one of these 37 diseases. That's your way of just trying to leverage the embeddings in the classifier head and seeing what disease they can predict. The other way to do it is just to use a language model. Just ask Quentry, just give it everything that you're giving to Bioreason, but in the ATCG will just be raw text and will just be passed on along with the question. And the model has to kind of look at the DNA sequence and try to do its best to answer. You can see that the main difference between these two scenarios is that the DNA model is really looking at the mutation. So it has the understanding of, does this mutation look like something that could cause disease or not, but doesn't really know the context of where that mutation is, what's the gene, what's the pathway, doesn't know any of those. And the LLM, on the other hand, has great understanding of what's the pathway, what's the gene. Maybe it remembers that these specific pathways are involved in certain types of diseases and it can make those correlations. But it really sucks at trying to look at the mutation and being able to kind of decipher what that mutation is doing because it hasn't seen mutations as much to be able to understand their effect. So these two kind of do modestly. Like you can see, the DNA models do like 86, 88%, and the Quen model, if go from 1B to 4B, goes from like 85 to like 93%. So we kind of see that they're kind of in the same level, but a Quentry 4B model can do decently well, but still far from being almost solving this benchmark. And then we have the Bioreason models, essentially combining them. Whether you take the nucleotide transformer or EVO2 with Quentry 1B or 4B, which gives you about like 4 different DNA LLM models. Now, if you look at the performance, you can see, for example, if you combine Quentry 4B with EVO2, you get a performance boost that goes to 97%. Like EVO2 itself is 88, Quen itself is 93, but when you combine them, you suddenly go to like 97%, essentially solving the benchmark. Or you can see similar scenarios with like nucleotide transformer, where even if you have like nucleotide transformer alone, it's getting like 86%, but once you combine it with the 4B model, it suddenly jumps to like 96%. And it's very interesting to see the performance boost you're getting, partially because you're bringing these two different understandings of biology together, and also because by bringing them together, you can do even better. Like 2 + 2 becomes 5, and you can even boost performance beyond that. And of course, we did also a bit of RL, just kind of showing that if you do RL, you can kind of boost performance a bit, like going from like 88 to almost like 90%. But the benchmark pretty much got saturated with just SFD, so it was hard to really show the full effect of what reinforcement learning could do here. Looking at like a case study is going to be interesting. So you can like look at a question. There has been like, another pathway involving actin and some other genes, and we have a mutation in profilin 1 gene, and we're going to ask what's the disease that it leads to. And you can see like the ground truth kind of makes sense as a biological reasoning, just goes from the disease, from the mutation all the way to the disease. But Bioreason reasons slightly differently. Like it's still reasoning correctly in biology, but it's slightly different of how you look at the problem and kind of argue that, yeah, I know that this PFN1 gene is profilin 1. I know profilin 1 is involved in like actin dynamics. And so if you mess up the actin dynamics, then you're going to mess up the muscle movements, which causes ALS and all those things. And so you can see it's slightly a different way to reason, still correct, but different from the ground truth. It kind of shows you that this approach has the opportunity to scale and actually provide novel insights into biology once trained enough. And the other data we also had was a bunch of variant effect predictions. In variant effect prediction, you have the same setup that there's some gene, there's some mutation, and you want to predict whether that mutation leads to a disease or not. But the main difference here is you don't really have any context on what's the pathway, what's the gene, what exactly is like the full biological context. You just know the exact gene name and where the mutation is happening and what's the sequences, and that's all. So you can't really see how it fits the bigger picture unless you have a good understanding of the gene name, but you don't really get the pathway. And we don't really know ourselves whether like how that mutation leads to disease. We just know whether it's pathogenic or benign. We don't really know why or how. These are stuff we've just got from MedLab, but we don't really have any idea of why that's happening. And so again, you could kind of set up the same task that you have a DNA model. We'll just do a binary classification on the embeddings of whether that will lead to a disease or not. And you could also have the language model kind of predict to you whether it's pathogenic or benign by just looking at the DNA sequence. Now, the performance difference between these two are quite nice. If you look at the DNA models, it gets like 60% or 70%. Like you can see that EVO2 is a superior DNA model, that nucleotide transformer. And that's because it's just trained on longer sequences, on more sequences, and also has a resolution of nucleotide level, which is more than the 6 nucleotide level of nucleotide transformer. But the language models suck. Like you're just getting 50%, which is pretty much random prediction. And so it's probably because like a language model can't really look at the mutation and decipher what's going on. It's very hard to do that when you haven't seen mutations in your training data before. All these variant coding mutations are like one nucleotide changing to something else. So you really have to understand the context of the DNA to understand whether that mutation is going to be neutral, it's going to have a big effect, or it's just going to be having some effect, but probably not going to be causing disease. And you can also, again, combine the DNA and language models together and see like what's happening. And you suddenly see that the same Quen model that was just doing barely 50% can jump to like 80% by just leveraging the DNA models embeddings. And this is quite remarkable because this gives like about 10 to 15% boost over the best baselines, here having been the DNA models themselves. So it's kind of showing you if you want to do variant prediction after you train a DNA model, just connect it to a Quen model and just have that do the predictions at the end, because it just gives you much better accuracy prediction. There's also the variant effect non-SNV data, kind of like same idea, but here it's not like non-SNV stands for non-single nucleotide variant, so it could be multiple nucleotides that are changing, or it could be like a deletion or something is removing, etc. And you can see that LLMs can do a bit better here because these are more mutations that if you look at like something is missing, probably you might guess that, okay, if you delete a nucleotide, it's going to be probably just causing a bad thing, so it might be pathogenic. And you can again see also the DNA models also do fairly well, but only when you connect them, you can go to like 89, 85% performance, which is much, much higher than all the baselines here. And so this kind of shows you that the approach works, not only just because it unlocks reasoning and kind of like leveraging low-level and high-level biology, but also just because maybe Quen tree can leverage the DNA embeddings much better than just any classifier head out there, kind of providing both the ML perspective of why this thing might work and the biological perspective, though I'm still very open to intuitions of why this thing works better than baselines. I would like to kind of summarize the contributions of this work as these 5 bullet points. I would say the main one has been creating this novel multimodal architecture. It's the first time that a DNA model is connecting to a large language model, and definitely not at the scale. Nobody has ever done anything. And so I think it's quite cool to see things coming together from the bio and the NLP side. There's, of course, advanced reasoning. It's the first time doing any bio reasoning, the new bioreasoning benchmarks like KEGG. I would say the best part is the empirical performance boost that you're seeing over the baselines. It could be even 10 to 15% within the short 50% range of variant effect prediction. And on top of all of these, you kind of get an interpretable reasoning trace out of like model thinking how the mutation is leading to a disease, which could be useful and kind of cool to look at when it makes mistakes or when it makes novel predictions. Our vision for BioReason is to create an AI system that can reason in biology from first principles. You can think of such system as being able to predict how mutations lead to disease, or how proteins functions, or how drugs could affect the cell state. All of these things are things that could be computed in biology, and you can reason from first principles, starting from the DNA and protein every sequences all the way to the end of the biology. And we believe that with the current setup and the architectures we have, given the right data and lots of compute, we can actually bring all of these things together and make a meaningful step towards some sort of biological superintelligence that can understand biology. And for us, the next steps have been just expanding the work. In BioReason 2, we've been spending a few months working on protein function prediction. Internally, we have state-of-the-art results on actually doing so. We'll be releasing this very much soon, either this month or next. We're working also on BioReason 3, which kind of works on now cell and kind of like reasoning across how a cell might change given some perturbations. And we've done some interesting partnerships across institute and some new institutes that have joined us through this last few months. And our team is it's about like 15, 16 people, with a few undergrads, a few PhD, a few scientists, and a few professors, just the breadth of everything you might have out there. And we're still expanding. So if you're interested to join us, let me know. Before I kind of wrap up, I would like to kind of summarize the sort of opportunities we have in the biospace right now. We can kind of break it down from three perspectives. One of them is the technology perspective of looking at the different ML approaches we're taking. One of them is like foundation models, right? We make DNA foundation models or protein foundation models or RNA foundation models. Just make a giant foundation model on something. Then the next one kind of becomes multimodality. Like how can you make a foundation model for all these modalities or bring these different foundation models together into a single model? This is going to be key because a lot of biology operates at the scale of gene and protein and RNA at the same time. So being able to understand all these different modalities together is crucial. Then you can also think of reasoning as a new technology that's going to be happening here and allowing us to go beyond what we've been able to do now. And ultimately, the agentic setup around these architectures could enable much better scientific understanding in these language models. If they can use bioinformatic databases or search up things or use different tools we use, then that would just allow them to be much better at biology, how biologists actually leverage these things. And the other side is kind of looking at the applications. You have the target discovery side of finding what protein or what target in the cell we should go and inhibit or activate or kind of deal with it to kind of help cure the disease. There's the drug discovery part that, okay, if you know that target, how do you find the molecule or design a molecule that's actually going to bind to it and cause that targeted effect? There's the protein design aspect that you can think of proteins as like these nanomachines, so they can do pretty much anything out there. And how do you design proteins with functions that only you have in mind and you want them to be able to carry those out? You can think about all the RNA therapeutics applications of how do we deal with this thing, virtual cell modeling, like kind of talking about if there's some perturbation, there's some drug or anything coming into the cell, how is it going to change the cell, how's the gene expression value is going to be changing. And so if we can pretty much model this within a virtual setting, then we can design drugs better because you can just go through hundreds of drugs and see which ones work best. And lots of application in genomics, from gene therapy to editing to all these different things we can do down there to help us understand and also maybe augment our genomics capabilities. And on the right, we have all the companies. There's like going to be a big space. Most companies right now are focused kind of in the drug discovery spec, from like isomorphic labs to Zyra to Deep Genomics, Recursion, Try Discovery. There's some companies think of like kind of making those bio or chemistry like intelligent models like Future House. There's like an RNA companies like there now, and then we have the big companies like OpenAI and DeepMind and NVIDIA putting lots of compute and money into these things. And of course, the pharma companies that don't want to left behind and want to kind of leverage this AI chance to find drugs themselves. And I guess between all of these things, kind of like the ARC Institute, which is the open source like bio research institute for kind of promoting the space and bringing researchers together to do cool things in biology that was not possible because of the scale of compute we have in academia. And I would like to kind of end with the quote from Jensen about digital biology. Jensen believes it's going to be the next amazing revolution. And the reason for that is because biology will now have the chance to be engineering, not science. When something becomes engineering, it kind of exponentially improves and compounds over previous years. You can also think of it when something becomes engineering, you can go and actually optimize for it. You can go solve it and design it. You won't use terms like drug discovery, you'll kind of use terms like drug engineering. You won't use terms like clinical trial, you'll use terms for clinical validation. You move beyond just treating biology as this very black box system and just doing trial and error to see what works, to a system that can actually be understood and just leverage to do any downstream thing for us. We can actually go and engineer, create, design, and do anything we want in this space, kind of like program it because of this super intelligent model that we have available that can enable us to finally understand biology. I believe it's going to be probably the biggest market out there in the future. And if you can truly unlock digital biology, a lot of things will change. We can solve all disease, we can solve aging, we can augment our capabilities, we can solve global warming, we can solve anything that kind of touches biology because all of these are understandable in the stack of science. And in case you're interested, just look at our project webpage. I think also made some viral tweet way back in June. And there's also our GitHub, which with all the data, everything available. And I hope that if you look at this work, you're going to be leveraging it and doing cooler works, kind of expanding the idea of bioreasoning within different places, with different data sets, with different models, and ultimately with different downstream tasks that are going to be all valuable. And this is my Twitter, in case you want to follow.
Good, thank you. Yeah, Samuel, go ahead.
Thank you, Adi, for the presentation. I myself is a computational biologist and working on neural network in specific part of the cell biologies. And although all the benchmarks and the work you've done has been very impressive and really have a strong capabilities in many use cases, I myself, like from the biology part, I have many questions actually, but it can decompose into two things. So one thing is that for DNA sequencing, over like 60-70% of the ATGC sequences are like repetitive. Like how do you train or how do you manage your data set to make it not like what kind of benchmark you use like to not like overfeed or like have a more contextual understanding with the variety of data. The second would be is that when we talk about genetic mutations, there are multiple stages of how DNA and RNA transcribe. So we have like transcriptomics, epigenomics, and proteomics, etc. And different omics data have a different approach on how to detect gene mutations or disease or dysfunctional part, etc. And I was wondering, like, does this model like have the capacity to work on like multi-omics data, or understanding different diseases or dysfunctional parts have a different requirement on how to understand these things? Like, so that's generally my two questions. Thank you very much.
Yeah, those are great questions. I'm going to first start with the DNA part. So first of all, when we train DNA foundation models like EVO2, we do train it on all sorts of DNA sequences. We train it on coding sequences, we train it on non-coding sequences, on bacterial sequences, human sequences. So it sees the whole breadth of what DNA could be, from the junk part to the actual coding parts, the differences between organisms and all of those things. And so after it sees all these different sequences, it will start understanding things across them that will give it very high quality embeddings for processing those DNA. Similar to how like a language model will just look at bunch of words in English and suddenly learn the language and understand the different principles in it. You can think of a DNA model also learning the same. By going through hundreds of billions of DNA tokens, you can understand the principles of genomics and understand how different things mean different things, etc. Here, though, we only use the coding sequence. We're just passing the actual gene sequence, and these are all in human. So we don't really have any junk DNA within the gene. We also don't use the introns, it's just the exons, just all together as the coding sequence. So everything there is super high quality. And the mutation is usually occurring, like the way we do it is we take the actual gene sequence. If it's too long, like if it's longer than 2048 as our maximum cutoff, then we'll have to truncate it a bit. But we kind of truncate around the window of the mutation. So the mutation is always there. Most of the time, the model will see the exact gene sequence, and sometimes we'll just see a part of it. But either way, it will see all the information it needs to see around the mutation to be able to kind of guess what's happening. And to your second question is about multi-omics. Yeah, I definitely think that kind of falls into the multimodality I mentioned. Like, bioreason as it stands right now can also only see DNA. And I would like to think of it as a proof of concept. Like, this thing needs to really scale from both the data size and the compute size to become something valuable that can actually be leveraged in the industry. But it's kind of showing you that in these small scale experiments, you're getting much performance boost. And you're kind of showing that different things that we didn't think is possible, like bioreason, could actually work. And so I think the ultimate form of something like this is when the language model goes just from being like a Q-entry 4B to like an actual 100B or 200B model, when you not only have just the DNA foundation model, but also RNA foundation model, protein foundation model, even a cell foundation model. Like, you have all these different foundation models. And the model is learned across diverse data sets to connect these modalities together and also leverage them to do reasoning. And such model now can easily do multi-omics, or can understand mutations much better from both the perspective of the mutation in the gene, also the change in the protein, to every other biological knowledge it has. And you can even argue that a 100B or 200B language model has way more biological knowledge than just a 4B Quinn model. So overall, it kind of seems that if you're at small scale, you're getting such great performance boosts. You can only do way better at larger scales, which is something we're pursuing. In Bioreason 2, we've scaled our amount of data. It's almost like, I think, a 10 or 100x amount of data. It's much more, it's like much more of an important biological task focused on proteins. And so I think it's going to be very interesting to see how that model now actually works, and it's being able to be leveraged to do interesting bioreasoning tasks on function prediction.
And you give an example here that for Parkinson's disease, or is there any other plan on work on other diseases or mutations?
So for Bioreason 1, we have 37 diseases. Most of it is Parkinson's, Alzheimer's, ALS, like this kind of like most well-studied disease, because you need the ground truths of the biologists to understand them. But we also have like a bit more rarer disease there. So it's like a diverse set of disease. And if you look at the performance, I don't have the graph here, but the performance is kind of consistent across all this disease. Like you get almost the same, like 97, 98 performance across all disease. Some of the disease you pretty much get all of it right, and some of them you might do a bit worse, but it's way more consistent than what I originally hoped for. So the model is kind of learning to still generalize a bit across this disease, but yeah, I think 37 is still a pretty low number. Ultimately, with like way more and more data, it could even do better, but we only have like these biologically verified ground truth only for this small subset of our data. So that's the only amount of data you have in KEGG to be able to train on.
Good. Any other questions? I have a question for you, Adi.
Yeah, go ahead.
I was wondering, so my background is in ML, know nothing about biology, but I find the DNA sequences interesting in the way that they are represented, because it's not normal text. It's just a bunch of 4 characters concatenated all over again. So did you experience any issues with that with respect to how DNA sequences are tokenized? And did you have to make any changes to the tokenization method of DNA sequences?
Yeah, that's a great question. So first of all, it's quite interesting what happens during training. So you can think of these DNA embeddings as kind of like alien tokens in the beginning, right? Like the adapter hasn't been trained, Quen hasn't changed much. So what happens is that the model will kind of look at them and be like, okay, I haven't really received the DNA sequence. That's the first thing we'll see. Then a bit later in the training steps, we'll say, I'm seeing some random like English or Chinese characters. I have no idea what they mean, right? It thinks like they're like kind of distorted embeddings of some words. And then it will suddenly like lose all its ability to think, and it will just put out gibberish. And then from there, it suddenly goes back up, and now it actually starts speaking language, and it's actually giving you some interesting biological traits. That's when the projector has been connected. And it's surprising that this happens very early on. Like with a few training steps, like I think it's less than like 10 or 5% of the training time, the projector is completely trained. Like it doesn't change much beyond that, and actually maps everything nicely. It was surprising for me because you kind of expect DNA and language to be very different from each other, right? Like if it's a vision language model, I can buy you by saying, okay, it's the same word, you know, it's the image of a cop versus the caption of a cop. So the model has kind of just map them quite nicely together and learn the projector for the vision side. But this is DNA. You really can't transcribe a DNA into natural language. You really have to kind of reason directly by paying attention to its tokens. So it was kind of remarkable for me to see how fast the projector connects. It kind of feels like when you train deep learning models at scale, they're really easily mappable to each other from the latent space perspective, which is quite nice because it helps us easily connect the DNA to the language model, but also makes you wonder whether the same trend could continue across all modalities. Like if you have foundation models for all modalities trained at scale and you bring all of them together, can they easily all learn to map and learn the same embedding representations in the latent space, kind of like a universal transformer latent space or something. And here, the way we tokenize data is we just follow the Evo2's tokenizer. We just say, okay, Evo2, just your DNA sequence, and it will just give us some embedding vectors, and we just smash those in the context of Quen. There's a bit of embedding engineering work to kind of make this work to be efficient and fast, because you need to do a lot of fast generations, but at the same time, like you can't add these tokens to the Quen trees tokenizer. This has to be tokenized and embedded by Evo2. So there's a bit of challenges there in the engineering side, but once you get them right, you can just pass in any DNA sequence of any length, and it will just add as many tokens that it needs to be able to carry out this task.
Okay, yeah, that makes sense. Thank you. And if there are any other questions, just raise your hand or unmute yourself. Feel free to ask. Otherwise, I have a few more questions.
I'm interested, like, contribute to this. Can you hear me? Sorry.
Yeah.
Yeah, I'm interested to contribute to this project. Is there a way to, I don't know, join your effort?
Okay, that's a good question. You can email me at this address here, or you can just like Google like the project or something and find my email. But this is my email, and just email me. We can have a chat afterwards to kind of discuss it.
Thanks.
Joanna, yeah.
Hi, can you hear me?
Yeah.
First of all, thank you so much, Adib. It was very insightful. I really enjoyed your presentation. I'd like to ask a bit about the interpretability of the results, because the domain is still a bit foggy for human researchers. There are a lot of things about DNA, genetics, etc., that we still don't know why, just that it does happen. I'd like to ask if there's a research towards using interpretability in reasoning models in biology as a way to help identifying the root causes of these things.
Yeah, that's a great question. And to be honest, one of the reasons we did bioreason was also because of interpretability. You can think of like look at the DNA foundation models, like EVO or nucleotide transformer. They are learning huge amounts of genomics. They've seen so much DNA sequences that they have understood a lot of things. But the only thing they can spit out is ATCGs. They're not really able to tell you as a human what they're understanding, what they're like learning. And so all that golden knowledge is kind of like locked behind this ATCG model. And so one of the reasons we did bioreason was to say, if you can take those embeddings out, can Quen kind of decipher them, like kind of like read them out and explain to us what the DNA model has been thinking along with its own understanding. And I think that's the best step to take here towards interpretability. I think that the ultimate form of interpretability is language. And I definitely think the reason bioreason is quite interesting is because you can look at the reasoning traces and kind of see if those make sense. Sometimes the reasoning traces don't make sense. Like the model is kind of like making up some biology to do it right. Sometimes they do make a lot of sense. Sometimes they're even in Chinese. Like there's so many different things that happens when you're training these things and you see so many different reasoning traces. But I ultimately think that's the better way to do interpretability. Of course, there's always this risk that at some point NLP will move from like doing reasoning on the tokens, like the language space, to maybe an embedding space. And I guess there you can have to kind of think something out. But I think this is the best way to bring out interpretability out of what a DNA foundation model has learned, apart from just looking at its entropies across sequences and trying to like decipher why those entropy values are changing and what they could represent. But yeah, I'm definitely open to like other ways you could interpret these models. Again, you'll still run to the same LLM interpretability issues of whether like this LLM is actually interpretable, which is something we face even in the natural language domain. But yeah, I hope this kind of answers your questions.
Yeah, absolutely. Thank you.
Yeah, I can see something in the chat. Neural networks leverage nominally quantum and post-quantum representations. Okay, that's a big title.
Yes, so like I wanted to write it in the chat, but like I forgot how enter doesn't create a new line but just send it, so I started sending a bunch of messages. Maybe I can ask the question out loud then. So basically that paper kind of like surprised me a bit, because what it does is that it takes a transformer based model and then it trains it on up to 4 billion tokens, and the tokens are from a set of 4 possible options, and they're basically the result of what they call a block random walk, and then they did some geometric representation, like that type of visualization. I think it's in figure 2, where they basically show, yes, in figure 2, but there's a better one in figure 3, where they plot the internal representations, and in the end they kind of see like a block sphere, basically for tokens that are generated through a random block walk. And I kind of like, I'm still struggling to get my head around that, because I'm having only like amateurish level of quantum understanding. But I was wondering, while you were presenting your work on DNA sequences, on like if a similar visualization would be done on the I think they did it on the activations, but I'm not sure, on the activations of the transformer you trained. Like, how would that look like for DNA sequences? Or and like, considering that you do have the transformers already trained, probably wouldn't be, or like, do you have them open weights? Did you publish them somewhere? Because I actually would like to try that, and it would be easier to get your models rather than train them from scratch.
Yeah, that's a good point. So first of all, I think this paper is quite interesting, but I have no idea what it means. So I have to kind of go and read it out. I'm not into quantum either. Like I'm a biologist who touched AI and then got into AI. But from the releasing model weight side, yeah, I think we should release it. Like we kind of held back a bit from it because on the one hand, we're a bit worried about safety stuff. Some people like messages, can we go and edit human genes with it? I was like, well, what are you going to use it? Like, I know it's not going to work, but kind of worried me. And then the second reason was because this was very inefficient. I think ultimately we got it working with VLLM, so it's actually fast in inference right now. But yeah, I think I'll plan to go and finish the last bit of it to be able to release the model weights. So that you can go and take a look. But essentially, the model weights for the Evo or nuclear transform is just frozen, and the projector is trained, and the Quen model is a bit lower. So the weights have changed a bit, but not too much. And I guess it's going to be interesting to like do these researches on it to see like how it shows. So yeah, like if hopefully I'll try to visit this month. We'll see how it goes.
Okay. Then I'm eager to see. I think the key to that one is that they're trained only on the tokens, and then they kind of get to compress the underlying process of generating those tokens in something that resembles symmetry and the fractalic nature of the block sphere. But if it's like if it's a language model trained on language and then fine-tuned on the genetic sequences, I think there's going to be lots of noise in the representation, but I would still be curious. I'm trying currently to play around a bit with other types of known sequences, but it's still work in progress.
Very well. Yeah, keep me in the loop. Kind of curious to know how it goes.
Okay, thanks. And thanks for the presentation. It was quite interesting to follow.
Yeah, thank you for listening. Any other questions? Yes, that's it.
Yeah, if there are, I have a couple last questions.
Okay.
Just very like overview of the project. But what was the most surprising result and finding when you were working on this?
I think finding that Guyanese is a token in the tokenizer of Quen was interesting for me. I have no idea what that means and why the model really liked generating that initially in its training before it actually spits out language. But I think the most surprising parts, yeah, one of them is definitely the fact that the projector trained so fast. I didn't expect the projector to be learned that quickly, because these are very different modalities. You kind of expect it to take it longer for it to learn, but it does learn surprisingly fast. The other thing that was interesting for me was the kind of additive boost that you get from having both of these things together. You can see that this one does like 10% better, that one does like 5% better, but once you combine them, you're starting to get like 30% better than like a random baseline. So it kind of shows you that there's some additive playoff going off between the DNA embeddings and the language model's understanding. It gives you a final better result than just combining them together in like an ensemble way or something. And I think the third interesting result for me was the kind of comparison, like going from Quen 1B to 4B boosted performance quite a lot. It's kind of like shows you that the bigger your language model and the more understanding it has, it can just be better. It's like a tiny scaling law experiment. So like we're just going from like 4B to 8B to 30B to 70B. I think it's going to be quite remarkable to see how it gets better.
Okay, and now that you're working on the version 2 and 3 of this model, what are the main advantages of these new models and the main surprising results that you're getting as you work on them?
So for Barzan 2, it was much harder to get this working. I think it's because like here in Barzan 1, we have a very low scale of data. It's still a proof of concept, so just you kind of just put it together and it kind of works, you know. But in Barzan 2, your goal is to beat the state-of-the-art models for protein function prediction, and like state-of-the-art has been developed for like many years. So being able to kind of like come out of nowhere and in 3 months kind of set this up together to be able to beat that, I think this was quite challenging. But we reached there, so I'm kind of excited to kind of release this, and you'll see the LLM god kind of like start reasoning what are the different domains in the protein and how they relate to its function just from looking at the sequence embedding. It's kind of cool. And for Barzan 3, I think the question now has become data. Like the data we want is super scarce. A very few amount of language combined with some biomodality data survives out there, especially if it has reasoning in it. And of course, like even for LLMs, we had the same problem. Like that's why like Scale AI survived as a company. But again, in biospace, we don't really have that ecosystem yet to have that data. And so I think it's quite challenging to get this research working without having that data. And you have to kind of generate it in-house and kind of leverage language models and biologists and know everything together to kind of combine it together. But I definitely think that the data is going to be the main moat here. And that's because human biologists can't really look at the sequence and reason. Like we can look at the mutation and be like, oh yeah, C to G change. I have a great idea what that means, right? and I think it's only the bioreason models that can do that. And so the highest quality data out there will be generated by these models, and then they'll be trained on that data to do even better next time. So I think here the kind of like the Scale AI and the OpenAI companies will not be different. They will be the same vertically integrated data model company. And that will be a strong moat because the best model generate the best data, which leads to the best models. And at some point, you'll take off and you'll be much better than anything out there. All right. Thanks for having me. Take care.
All right. See you all.